Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we are beginning Chapter 1, an overview of recombinant DNA technology. This chapter introduces the central logic behind modern genetic engineering, how scientists identify useful DNA, combine it with a suitable vector, move it into a compatible host, and prove that the resulting system works as intended. By the end of the lesson, you will be able to explain the complete recombinant DNA workflow, connect every molecular tool to its purpose, and distinguish a likely recombinant from a scientifically verified result. Keep your notebook ready, pause whenever you need to study a diagram, and let us build the chapter step by step. Here is everything we are going to learn. We will first define recombinant DNA technology and examine the scientific foundations that made it possible. We will follow the complete gene-to-clone workflow, from identifying and obtaining an insert, to choosing a vector and host, joining DNA and introducing the construct into a cell. We will compare genomic DNA, complementary DNA, PCR-derived and synthesized inserts. We will study restriction enzymes, compatible DNA ends, and DNA ligase, then connect the origin of replication, selectable marker, cloning region and expression signals to their functions. We will learn why host biology matters and clearly distinguish selection, screening, and confirmation. We will separate DNA cloning from gene expression and organize the evidence levels of DNA, RNA, protein, and biological function. We will apply the workflow to recombinant insulin, explore medical, agricultural, industrial, and research applications, and examine biosafety and ethical responsibility. Finally, we will practice organizing diagrams, flowcharts, and explanations for full-mark CBSE answers. With that roadmap in mind, let us begin with the central question of the chapter. Imagine that a living cell is a tiny factory, and DNA is the instruction library inside it. Now suppose one organism has a useful instruction, perhaps the instruction for making human insulin, and we want a safe, controllable host cell to use that instruction. How could we identify the right DNA, move it, copy it, and prove that it still works? That is the central problem solved by recombinant DNA technology. But here is the important part. This chapter is not a vocabulary list. It is a chain of decisions. Every tool has a purpose, every stage has a test, and every successful application depends on evidence. Keep one question in your mind throughout the lesson. What must be true before we can confidently say that a recombinant DNA experiment has succeeded? Recombinant DNA technology is a controlled set of molecular methods used to manipulate nucleic acid directly. A selected DNA segment called the insert is joined to a carrier DNA molecule called the vector. The resulting recombinant DNA is introduced into a suitable host cell, where it may be copied, studied, or expressed. Notice the word controlled. DNA recombination also occurs naturally, for example during meiosis. But laboratory recombinant DNA technology has defined source material, deliberate molecular steps, and a planned outcome. That outcome might be many copies of a gene, production of a protein, introduction of a trait, or construction of a research model. A complete definition therefore includes 4 ideas: the DNA segment, the vector, the host, and the purpose. Quick check. If 2 DNA molecules join naturally during meiosis, is that automatically recombinant DNA technology? No. The laboratory workflow and intended manipulation are missing. No single discovery created recombinant DNA technology. Genetics connected genes with inherited traits and helped scientists identify useful candidates. Molecular biology explained how information moves from DNA to RNA to protein. Biochemistry supplied methods for isolating nucleic acids and enzymes that cut, copy, and join DNA. Microbiology provided fast-growing, genetically understood hosts such as laboratory strains of Escherichia coli. Cell biology later expanded the possibilities to yeast, plant, and animal cells. Analytical methods made it possible to inspect the DNA and protein produced. In an exam, do not write only, many branches of biology contributed. Connect each branch to an actual job. Genetics identifies the target. Biochemistry supplies molecular tools. Microbiology supplies the host. Analytical biology verifies the outcome. That cause and effect structure turns a vague answer into a scientific one. Let us build the complete workflow. Step 1. Define the biological question and identify the gene or DNA sequence of interest. Step 2. Obtain that sequence as an insert from genomic DNA, complementary DNA, PCR amplification or synthesis. Step 3. Choose and prepare a vector with the features needed for replication and selection. Step 4. Prepare the vector and insert, then join them to form recombinant DNA. Step 5. Introduce that construct into a compatible host by transformation or another delivery method. Step 6. Apply selection, reducing the population to cells with the required vector-associated trait. Step 7. Screen those survivors to locate the desired recombinant. Step 8. Confirm insert identity, orientation and sequence. Only after confirmation do we propagate the clone or test expression and function. Pause here. Why is confirmation placed after screening? Because screening produces likely candidates, not certainty. The logic of the chapter is a narrowing funnel. Many cells, fewer survivors, fewer likely clones, and finally a verified construct. The insert is not merely the gene. Its source and boundaries matter. Genomic DNA contains the sequence as it exists in the chromosome, including introns in many eukaryotic genes and surrounding regulatory regions. Complementary DNA or cDNA is produced from processed messenger RNA, so it represents a gene that was expressed in the sampled cell and usually lacks introns. Why is that useful? A bacterial host generally cannot remove eukaryotic introns. If we want bacteria to produce a human protein, cDNA may therefore be more suitable than the complete genomic gene. PCR can amplify a defined region, while chemical synthesis can produce a designed sequence. The insert must also preserve the correct reading frame and any required regulatory or signal information. Prediction question. For bacterial production of a human protein, would you usually begin with human genomic DNA or cDNA? cDNA is often the better starting point because the introns have already been removed during RNA processing. Restriction endonucleases recognize particular DNA sequences and cleave phosphodiester bonds in the DNA backbone. Some create overhanging sticky ends, others create blunt ends. If vector and insert are cut to produce compatible ends, complementary bases can pair temporarily. DNA ligase then seals breaks in the sugar phosphate backbone. Think of restriction enzymes as precise molecular scissors and ligase as the enzyme that completes the backbone. But do not stretch the analogy too far. Ligase does not know which product we want. A vector may close without the insert, and an insert may enter in the wrong orientation. Using two different restriction enzymes can support directional cloning, yet the resulting colony still requires screening and confirmation. Exam trap number 1. Never say restriction enzymes join DNA. Exam trap number 2. Never say ligase selects the desired gene. Cutting, joining, and identifying are three separate jobs. A useful vector is an engineered DNA system. The origin of replication allows the vector to be copied in a compatible host. A selectable marker gives cells carrying the vector a trait that can be selected under defined conditions. A cloning region provides convenient sites for inserting foreign DNA. If the goal is protein production, an expression vector also needs a promoter recognized by the host, appropriate translation signals, a correct reading frame, and often a termination signal. Some vectors include tags or secretion signals to help purify the product. Here is a powerful way to remember vector features: replication, recognition, and result. Replication asks, can the vector be copied? Recognition asks, can cells carrying it be selected and recombinants identified? Result asks, can the intended gene be expressed correctly? A circular drawing with labels and functions is far stronger than a list of vector parts without explanation. The host is not an empty container. It supplies replication enzymes, transcription machinery, ribosomes, energy, and raw materials. Escherichia coli is popular because it grows rapidly and many laboratory strains are well understood. But speed is not everything. A human protein may require folding, cleavage, or chemical modifications that bacteria cannot perform properly. Yeast can provide some eukaryotic processing. Plant and animal cells may reproduce specialized modifications more accurately, but they are usually slower and more demanding to culture. Host choice is therefore a biological decision. Ask 3 questions. Will the vector replicate here? Will the host recognize the expression signals? Can the host produce a functional product? A construct that is copied successfully but makes no active protein may have a promoter problem, a reading frame problem, a folding problem, or a host processing problem. Presence of DNA is only the beginning of the evidence chain. After delivery, imagine a plate containing many cells. Some received no vector. Some received an empty vector. Some may contain the insert in the wrong orientation. Only a few may hold the exact construct we want. Selection applies a condition that favors cells with a chosen vector-associated marker. It removes many failures, but empty vector cells may still survive. Screening then examines selected colonies using a method such as insertional inactivation, colony PCR, probe hybridization, or diagnostic restriction digestion. This produces likely recombinants. Confirmation provides stronger molecular evidence, often including sequencing of the insert and junctions. Keep the questions separate. Selection asks, which cells carry the selected trait? Screening asks, which survivors are likely to contain the desired recombinant? Confirmation asks, is the construct molecularly correct? If you remember only one exam distinction from this lesson, remember this 3-level evidence ladder. Gene cloning means producing many copies of a DNA sequence by maintaining a recombinant molecule in a host. Gene expression means using that sequence to produce RNA or protein. These are different claims, so they need different evidence. A restriction digest, PCR, or sequencing result can confirm the insert at the DNA level. RNA analysis can test transcription. Protein detection can show that a product of the expected size is present. But even that does not prove biological activity. A functional assay must test what the protein actually does. This creates an evidence ladder. DNA, RNA, protein, function. Each level answers a new question. Suppose a bacterial clone contains the correct human gene, but no active protein is detected. The cloning may have succeeded, while expression failed. That single distinction explains why good biotechnology never relies on one test for every conclusion. Let us apply the workflow to recombinant human insulin. The goal is not simply to place human DNA in bacteria. The goal is to obtain a safe, active hormone. Because bacteria cannot remove human introns, an intron-free coding sequence derived from processed RNA or a designed sequence is used. That sequence is inserted into an expression vector with host-compatible regulatory signals. The construct enters a production host, and selected colonies are screened and sequence confirmed. Expression conditions are then adjusted so that the insulin chains or precursor are produced. The product must be recovered, processed correctly, purified and tested. DNA confirmation proves the construct. Protein analysis supports identity and purity. A biological assay supports activity. Manufacturing tests address consistency and contaminants. Notice how the application contains the same chapter logic, but extends beyond cloning. A recombinant product is not ready merely because a colony grows. It moves through a chain of molecular, biochemical, and quality evidence. The recombinant DNA platform is powerful because the workflow can be adapted to different genes, hosts, and outcomes. Health applications include therapeutic proteins such as insulin, selected vaccine antigens, antibodies, and diagnostic reagents. Agricultural applications may involve pest resistance, nutritional traits, or responses to environmental stress. Industrial hosts can produce enzymes and other biological molecules at scale. Researchers clone genes to investigate regulation, mutation, and protein function. Yet every field adds its own evidence. A therapeutic protein requires identity, purity, potency, and safety testing. A transgenic plant requires molecular confirmation, stable inheritance, expression, and agronomic evaluation, followed by relevant environmental and food or feed assessment. An industrial process requires yield, consistency, and quality control. The reusable principle is construct, confirm, test function, and then validate for the intended application. Responsible recombinant DNA work begins with risk assessment, not after the experiment is finished. Which host is being used? What sequence was introduced? Is a biologically active product expressed? How could exposure occur? What might happen outside the intended setting? Controls may include disabled laboratory strains, physical containment, careful work practices, decontamination, and documented disposal. Ethical questions may involve informed consent, animal welfare, environmental consequences, ownership of biological material, fairness of access, and honest public communication. Biosafety does not mean declaring every recombinant organism dangerous. It means choosing controls that match the organism, construct, and exposure pathway. Ethics does not replace science, and science does not settle every ethical question. A mature answer shows both evidence-based risk control and responsible consideration of people, animals, and ecosystems. For a 3 or 5 mark CBSE answer, begin with the precise definition. Then present the process in order: candidate gene, insert preparation, vector preparation, joining, host entry, selection, screening, conformation, and finally propagation or expression. Beside important tools, state their function. Restriction enzymes cut at recognized sequences. Ligase seals the backbone. The origin supports replication. A selectable marker supports selection. If the question asks for an application, connect the construct to the actual product or phenotype and mention the required validation. For a diagram, use a title, large recognizable shapes, directional arrows, and relevant labels. Avoid 3 common errors: treating selection as proof of the insert, confusing cloning with expression, and listing benefits without quality or safety evidence. Structure creates marks because it makes your biological reasoning visible. Close your notes for 1 minute and rebuild the chapter. A useful DNA sequence is obtained as an insert. A compatible vector is prepared. Molecular tools join insert and vector to create recombinant DNA. The construct enters a suitable host. Selection removes many cells that lack the chosen trait. Screening locates likely recombinants. Conformation establishes the correct construct. The clone is propagated, or the gene is expressed and tested at the RNA, protein, and functional levels. The result can then move toward an application with appropriate quality, biosafety, and ethical review. Final challenge. Question 1. Why can selection not replace screening? Question 2. Why is cDNA often useful for producing a human protein in bacteria? Question 3. Which evidence proves cloning, and which additional evidence supports functional expression? If you can answer all 3 in complete sentences, you have moved beyond memorization to conceptual mastery.